Hi, my name is Zoha, and my presentation is on siRNA or cRNA. So let's talk about the introduction and background of cRNA. One of the classes of small RNA molecules is called cRNA, and these are small interfering or silencing RNAs that regulate the expression of specific genes. And they do it by a process of, or by silencing the process of transcription or translation of genes by binding to the complementary sequences in distinct messenger RNAs. And these are, because they are just smaller versions of regular RNAs, they look exactly the same, meaning they are double-stranded, but they are just shorter. And here's an image of how a double-stranded RNA is turned into a cRNA. And the first step, there's different steps, but I'm just showing here the step where the RNA turns into cRNA. And it says a dicer cleaves double-stranded RNA into a cRNA. And it also has about 21 to 22 base pairs in length. So, an application of cRNAs that is used mostly and widely is in the field of therapeutics and nanomedicine because of their ability to silence targeted genes. Of course, there are major limitations, but cRNAs have been used for treatment of diseases and also the augmentation of tissue formation. And the cRNAs are used in therapeutics, and it's about 3 steps. And it begins with the design of cRNA that is made, that is, it binds to the proteins in our cells or tissues that have that disease or cancer cells, if you can say. That would be the first step. The second step is the incorporation of the cRNA into a delivery conveyance that can effectively convey the cRNA in vivo. So basically, the cRNA is not yet inside the cell, but the process of getting it there would be the second. And the third and the final step would be the delivery of the cRNA to the designated site or the tissue or the cells where the gene silencing will take place. So that would be the last step. And that's pretty much the application. Um, I am not going to get into detail because my article does not go into detail. And this is the general information that I found on the cRNA, because the 3 steps are same for any application of cRNA, because ultimately what it's doing is silencing the gene, and it can only be done um, in a few different areas, but since we're talking about cRNAs, it is pretty much the same for all of them. So these are the 3 steps that are going to be used to silence the genes. So here I'm going to talk about a couple different things, starting with my key figure from the book actually. I found this in vivo therapeutic success little paragraph, and then there is a table underneath, which is the table 4 in the book. It's a long table, but I don't have space for all of the things, so I just put one example from that table. So let's talk about the therapeutic success. Here it says that um, the clinical studies of for using cRNA for therapeutic gene silencing are currently underway, and in this case, there was a successful case of cRNA transfer, which was targeted VEGF for the treatment of wet neovascular age-related macular degeneration, which is basically when people get older, they start losing their sight, which is macular degeneration, and it can be resulted from different things. But this treatment of CRNA delivery was done, and it showed pretty significant results, and they were very promising. But this is just one case scenario. This treatment, I guess, is done widely, but people know about it, but it's not something that's done like every single day. So it's not something that people are even familiar with. And it can be done with different things, but from what I found, it's still a process of experimentation. So here there is one example that you can see for the target, which was the disease. It was the heart, and the disease is right there. It's restenosis, and so what happened was within the heart tissue or the cells in the heart, there was gene silencing that happened through CRNA, and here is the CRNA sequence present over here, and also the name of the targeted genes, as you can see. So that's just one example, but there are several in this table of different organs in the body, and that this can be done basically anywhere in the body. So for my question, my actual question does not reflect what you see on the screen. I made a mistake. So my original question was, what is the difference between the application of CRNA and genetic modification, not gene sequencing? So basically, genetic modification is a form of CRNA, I think. Um, but is it the same thing? Is it completely different? Or are they just, different names of the same thing? I know genetic modification can be adding a gene, deleting a gene, things like that. And for CRNA, it's silencing the gene. But so does that mean that CRNA or application of CRNA is also genetic modification? I just need some clarification on that. Thank you.